everyone, my name is Dr. Valerie Meyer-Spiridolozzi, and I teach Industrial and Systems Engineering here at the University of Rhode Island. The Yukon URI Navy STEM Coalition works with the National Institute for Undersea Vehicle Technology and many of our partners throughout the region who build submarines and support our defense industries. Come take a look at what we're doing.
Hi everyone, my name is Lauren. I'm a senior at the University of Rhode Island. Today we're at Dr. Roxbury's NanoBio Engineering Lab, where we do a lot of work with carbon nanotubes. Let's take a look at some of the research we're doing. All right, welcome to our cell culture room. This is the place where we culture and grow all of our cells that we perform experiments on with our carbon nanotubes. We heard that you've been learning a lot about cells in your science classes. What is a cell? What are the different kinds of cells? So a cell is a basic unit of life. It's what makes up everything that is living. There are many different types of cells. In our lab specifically, we use animal or mammalian cells to do all of our experiments. So my question is, what do cells need to live and grow? That's interesting. In our lab, we keep cells alive using something called cell media. This is what it looks like, and it contains a variety of salts, sugars, amino acids, and antibiotics. And we add this to all of our cell cultures to help keep them growing and thriving. Now, how would each of the components, being salt, sugar, amino acids, and antibiotics, help the cells to live and grow? Along with our cell media, here's some of the other equipment that we use when we work with our cells. I don't know, you might recognize some of these materials. This, for instance, is our pipette. This one specifically can get measurements all the way down to 2 microliters. This is the cell culture hood. We have to work underneath this hood whenever we do any cell culture experiments because it keeps everything sterile. Anything that I'm going to use underneath the hood, I need to spray down with ethanol first. Ethanol is the same ingredient that's found in hand sanitizer. The first thing I'm going to do is suction out this old media. This cell line in particular, all of the cells adhere to the bottom of the flask, so I don't have to worry about suctioning out the cells by accident. The next thing I'm gonna do is using another one of those pipettes. I'm gonna wash away any extra debris or dead cells that might be still on the flask. And again, I'm just going to suction it out. Now that we have a relatively clean flask, just full of cells, I'm going to add just a little bit of cell media. This is 1 milliliter and a 2nd milliliter. In order to get the cells off from the bottom of the flask, I'm going to use a cell scraper and very simply just move it back and forth across the bottom of the flask, hopefully to pick up all of the cells that are stuck on there. Now that we have a solution of our cells, I'm going to prep the new flask. This is just done by adding about 17 or 18 milliliters of that cell media to the new flask. And the final step is to take a fraction of this old cell solution and add it into our brand new flask. And this will allow our cells to continue to grow and be healthy and live out the rest of their lives. I'm gonna add about 450 microliters. Before I do that, I'm just gonna make sure to do something called pipette mixing up and down, and this will help to make sure there aren't any clumps of cells that we're transferring over to the new flask. Now that we have a new flask with fresh media and fresh cells, I'm just gonna shake the flask back and forth a little bit to evenly distribute the cells among the whole bottom of the flask. And the last step is always to label the new flask. Along with the date and my initials, I'm also going to label what cell line this is, which are our raw cells, and finally the passage number. which is just the amount of times that we've done this process with the cells. So this is passage number 18. And here we have a fresh flask of new cells. And over here is our cell incubator. What environmental factors do we need to consider to keep our cells growing? Now, this incubator incubates our cells. It keeps them nice and warm at about 37 degrees Celsius, and it also provides enough carbon dioxide for the cells to live. But why do you think we're using cells in our experiments? We're using cells in our experiments because we're trying to see how carbon nanotubes respond in living environments. And using cells is perhaps the easiest way to do this without injecting carbon nanotubes into multicellular beings. Do you recognize this tool? This is our microscope, and I use it anytime I'm about to work with my cells. It allows me to see the cells really close up before I work with them. What parts of the microscope do you recognize? And what are these parts used for? This is the stage where I'll put any of my samples, and then we have the fine adjustment knob and the coarse adjustment knobs. Both allow us to focus our field of view. And at the bottom here, you have our objective lenses, and this allows us to magnify the image to a certain extent. On this compound microscope, the max magnification that we can get to is 40x. So now that the cells are all prepped and ready, we're able to run our experiments on them. So this can look like adding or dosing the cells with our carbon nanotube samples. We're able to image our cells with the nanotubes inside and see the nanotubes fluoresce in responses to different environments, or just over time to see where the nanotubes are inside the cells. Here we are in the microscope room. Here in the NanoBio lab, we have a specialized near-infrared microscope, and this is able to see wavelengths of light just outside of what our human eyes can see. Today, I'm going to be running experiments on breast cancer cells that I have growing in these two little petri dishes. I'm going to image them using this microscope, and we'll be able to see some carbon nanotubes fluoresce. First thing I'm going to do is put the petri dishes inside a little stage incubator that we have right here. Just like our big incubator that we have in the cell culture room, this allows us to set a certain temperature and carbon dioxide level for the cells. Now that that's all set, I will turn on the microscope. And today I will be using a 100x objective lens. Because the 100x objective is a high-powered objective lens, we will just use a little drop of oil on the top in order to get that image that we need. With that all set up, I can put the cells on the stage. And I'm going to position the stage so that it's right over our objective lens. Now that everything's all set up, we are ready to take images. This is a white light image of one of the cells. Sometimes when you take the fluorescent images with the laser, it's actually easier to tell where the nucleus might be because there'll be like no carbon nanotube fluorescence coming from that spot. The glowing dots you see there are carbon nanotubes. I always love the way carbon nanotubes look because it reminds me of the stars in the sky. The carbon nanotubes and other fluorescent particles will absorb a certain energy, a certain wavelength of light, and then emit a different wavelength, a different energy of light, and that's what we're able to see. Depending on the environment, a carbon nanotube's fluorescence will change. So for example, in this study, I can expect that working in a cancer cell, like the breast cancer cell I have here, the fluorescence will look different if it was in a non-cancerous cell. If you had access to any of this equipment here, how would you use it to help your community and those around you? For me, I always wanted to be a doctor or work in the medical field because I thought that was really interesting. I loved science and math in middle school and high school. Once I got to college, I realized that engineering was the perfect realm for me because I loved the problem-solving aspect. I didn't necessarily want to be working with patients and other doctors, but I wanted to be an engineer solving the problems kind of in the background. Where I am now in this lab and hopefully in the future, I'm hoping I can work on new medical technologies, drug discovery, and other biomedical applications to help out the world. If you have any interests like that, definitely consider checking out the programs at URI, chemical engineering, biomedical engineering, and research in general.